All right, ladies and gentlemen, we are going to try some new technology. It's a new computer, so you won't see anything different, and see if it will let me record and write notes. So what we're going to talk about is the inheritance of traits. So before we go any further, let's do a mini review of genetics from your freshman year. So there's an idea in genetics called the central dogma of genetics. It's kind of one of the most important central themes. And that is that you always start your information carrying with DNA. That message then gets transcribed into RNA. It actually passes through two different RNAs, through mRNA and then tRNA. Then that ends up being translated and coding for a protein. And then when that protein is produced, that creates basically a trait. So we're going to refer to these processes as process one is transcription, process two is translation, and we're going to say process three is expression, even though technically, like making a copy of the gene is expression. This sequence is the same in every single organism on the planet, from bacteria all the way to humans, to snakes, to whales, to plants, to fungi, to protists. All of these organisms follow the same pathway of information, from DNA all the way through to an expressed trait. And what we're going to focus on right now is the trait part of this. Later, we're going to talk about the intricacies of DNA. We'll talk about mRNA, transcription, translation, and how proteins are produced, but that will come in our next unit. So this is just to kind of reinforce that. So we have a prokaryotic cell that is performing the process of protein synthesis. Notice it's got DNA, it's got RNA, it's got ribosomes, and those ribosomes are used to make the protein. In a eukaryotic cell, we have some of the same stuff. We have DNA, we have RNA, then we have ribosomes, which are used to make protein. So the same basic structures are there. There's some differences in complexity. There's also the fact that in eukaryotes you have a nucleus, but all of the same components are present in the cells. I'm going to go over a few terms just to make sure we remember what they mean. So the genome is all of the genes in an organism. You can visualize it with something called a karyotype, which is basically a picture of all the chromosomes during mitosis. They're the condensed chromosomes so that you can actually see the shapes, and then you can apply various dyes or do artificial coloring. So what you see on the left is a karyotype for a human, and you can see I haven't counted them, but it should be all 46 chromosomes there. On the right-hand side, what you see is a fruit fly genome with the 4 chromosomes that make up the fruit fly genome. Let's also remind ourselves what alleles are. So an allele is a particular variant of a gene. So depending on what you inherit from your parents, you might have one version of a trait or another version of a trait. The trait that's most common in most of the population is called the wild type allele. And you get new alleles basically created by mutations in your DNA. So let's look at a wild type tiger. This is the wild type tiger. That is what most Bengal tigers' coloration looks like. There is a mutation at the allele that codes for this brownish orange color, and when it is not working, you get a Siberian tiger, which is what the middle image is. And then you can have a mutation at another locus or location, which actually stops any pigment from being expressed, including the black stripes, and that gives you an albino tiger like you see here. Genes that have more than one allele possible are called polymorphic. So a big example of that is going to be blood type. That gene can code for either what's called an A antigen, a B antigen, or it can code for not having that antigen at all. So there are 3 possible alleles that you can have at that genetic location to give you your different blood types. More terminology. Remember, a dominant allele is the one that's always expressed. in the phenotype of an organism. You will always see it in the organism, and we represent that with capital letters. Recessive alleles are only expressed when there is no dominant present, and we represent that with lowercase letters. My apologies, I copied and pasted without looking, but that's supposed to be lowercase letters right there. If you have two copies of the same allele, you are homozygous, and since you can be either have both dominants, like so, or both recessives, like so, you can be either homozygous dominant or homozygous recessive. You do need to specify because it does make a difference. So when you're writing an FRQ, or if you're talking to a professor, you'll want to use the terms homozygous dominant and homozygous recessive. Then if you have two different alleles, you are heterozygous. Keep in mind, there's no heterozygous dominant, heterozygous recessive, because for our simpler genetics problems, if you're heterozygous, you're always going to have one dominant and one recessive. When you're doing this, no matter what kind of genetics problem you're doing, you always write the dominant allele first. So you should never write your heterozygote like that. Run the risk of it being counted wrong. With traditional complete dominance, a heterozygote or a heterozygous organism always will show the dominant phenotype. So I've mentioned genotypes and phenotypes a couple of times, so let's talk about what those are. The genes are the actual DNA sequences that you have that are going to ultimately code for a protein. Different versions or different alleles will code for slightly different versions of proteins. So what we have up here at the top, we have 3 different genotypes: homozygous dominant, heterozygous, and then we have homozygous recessive. Based on the genotype or the genes you have and what traits you express, you will have different phenotypes. So here, for complete dominance, you only have 2 different phenotypes. You have round, for this example, and you have wrinkled. So when you look at this at a genetic level, we can say that our round through experiments has been determined to be dominant, and our wrinkled through experiments has been determined to be recessive. In a lot of cases, what will happen is a dominant gene will code for a fully functional protein, and a recessive gene will code for a lot of times a protein that's non-functional, has reduced function, or in some case will just prevent expression of that. So if we're looking at an example here of this round and wrinkled peas, you can actually trace that trait to a specific gene. And that specific gene is a specific enzyme that's necessary to make starches. Without that enzyme, you don't make starches properly, which changes the shape of the pea. And it's actually just due to a weird insertion mutation that adds in 800 nucleotides. And that changes the shape of the protein, which changes its function, which then changes the shape of, a body part. We're also going to need to refer to generations. You will see this terminology if you see any diagrams on the AP test looking at genetic crosses. You'll also see them if you see any pedigrees. So when we're talking about what we call a cross, which is looking at the offspring of 2 different individuals, the first cross that we look at is the parental generation, also labeled the P generation. Every generation after that is going to have F in the title. Why? Because they are filial generations. Filial comes from the word for son. So they're the offspring generations. So your first filial generation, the first generation of offspring, is referred to as the F1 generation. In kind of the really typical crosses that were performed, for example, by Gregor Mendel and some of those examples, your F1 generation will often be hybrids, meaning they're heterozygotes, because you will get kind of purebred or true or homozygous individuals for your parental generation. And then usually you'll also do a second filial generation, so an F2 generation, and look at the offspring of 2 of those hybrids producing or offspring with each other. This is pretty simple to do in plants because you can have plants self-pollinate. It's a little creepier with animals when you're doing experiments like this, or when people are doing like dog breeding or animal breeding. Sometimes they'll mate brother and sister, a very close relation, just to make sure that they get the desired genes. So we left off on our last lesson with meiosis. Let's talk a little more about the process of fertilization. So let's say we start with this set of chromosomes. We've got n equals 3. It's diploid, so 2n equals 6. And I have 3 purple chromosomes and 3 green chromosomes, just to represent the fact that we have homologous pairs. So we're going to label these as chromosome 1. The next larger ones will be chromosome 2, or chromosome pair 2, and then the smallest ones will be chromosome pair 3. When I refer to them, that's what I'm talking about. Now, let's say we're looking at a cell in meiosis. During meiosis 1, what's going to happen is these chromosomes, these homologous pairs, will line up along the metaphase plate. And we're going to represent that with that kind of gray square there. After metaphase 1, when these start to separate, they will move apart from each other. And what we're going to pull up, we're going to look at, is the possible gametes that are made. So in that with that first set of chromosomes that are going to the left, you'll have a gamete that will get these 3. With the chromosomes or the homologs on the right, the gamete will get these 3 chromosomes. So you make a potential gamete that has all purple chromosomes. You make a potential gamete that has all green chromosomes. But that's not the only way that these chromosomes can line up along the metaphase plate. So for example, we have a different arrangement here. If we look, it will produce different gametes. So this gamete right here is different from either of the 2 gametes that were produced with our first potential meiosis run. Then we can have yet another orientation or arrangement, which will produce yet more arrangements of gametes. And we can still have even more. Now, there are actually more combinations possible, but I ran out of room on the slide. So we're just going to look now at, let's say we have one particular sperm fertilizing one particular egg. Each of these are haploid. So each of them has one chromosome from each homologous pair that we can have, and altogether 3 chromosomes. After fertilization happens, you'll have all of these chromosomes now present in one nucleus. So now you have restored this back to 2n, or diploid. So remember, 2n is 6. And the reason why we don't see those X shapes is because these are unduplicated chromosomes. There are no sister chromatids. And this cell is now able to divide and eventually make a full-on organism using the genes in these 6 chromosomes. Now what we're going to do is we're going to look at how that relates to inheritance. And we're also going to look at Gregor Mendel's laws. So he had 2 major laws that he kind of came up with after the results of a lot of different experiments. And this has been refined over time as we have learned more and more about the genetic material and how DNA is packaged and chromosomes and all of that. So genes are packaged on chromosomes. Each chromosome contains specific genes. So for example, what you see here is a map of some traits on the X chromosome in humans and a map of some traits on the Y chromosome in humans. These are not by any means all of the genes on a chromosome. I looked at some chromosome maps, and if I'd found one with all of the genes, it would have been very difficult to read and really confusing. But when you have meiosis happen, each chromosome of any given homologous pair is inherited independently of the others. So you could get, for example, these different arrangements. So you don't necessarily have all of your purple chromosomes in one gamete or all of your green chromosomes in one gamete. those chromosomes sort independently of each other. That is the law of independent assortment. Basically, each chromosome or each gene in what Gregor Mendel was studying sorts independently of the others. So there's no relationship between them. Now, this only holds true when you have genes on separate chromosomes. If you were looking at genes on the same chromosome, those would actually be linked with each other. That's a more complicated idea that we may not get to this year, but it's something you'll learn more about in genetics. There's also the law of segregation, where during meiosis 1, when homologous chromosomes separate, that separates the two copies of a gene that each individual has. So each gamete is only going to get one copy of a gene. We also are going to start talking about probability. So if you've been enjoying our mathless time, sorry, that's not going to continue. Any event that has an 100% probability of happening, it's guaranteed to happen, has a probability of 1, or you can express that as a percentage and say it's 100%. An event that is impossible, that absolutely cannot happen, has a probability of 0, or once again, you can say it's got a 0% chance of happening. Any other events that could happen but aren't guaranteed to happen have a probability somewhere between 0 and 1. And you can express this as a fraction, so a 1 in 2 chance of happening or a 1 in 4 chance of happening. Or you can express this as a decimal, a 0.5 probability or a 0.25 probability. Or you can express this as a percentage, so a 50% chance or a 25% chance. All of these are the same. They're expressing the same thing. Half of the time, this outcome is going to happen. So let's look now at probability with inheritance. So the chance of any given chromosome out of a homologous pair being passed to a gamete is 1 out of 2. That means that you have a 50% chance of passing on one particular allele to your offspring. Now we're going to start introducing those letters. So let's say that our purple chromosome has a dominant trait, a dominant allele, and our green chromosome has a recessive trait, a recessive allele. What that means is that one of our gametes has the dominant trait or dominant allele, and one of our gametes has the recessive allele. And this becomes important when we start looking at how genes are distributed. Basically the same thing as the previous slide, but now I have taken out the pictures of the chromosomes, and instead of representing chromosomes, we're now going to represent genes using these letters. That way we can show dominant and recessive. Apologies, there's a cat fight going on. So this brings us to something that you used quite a bit when you were a freshman, Punnett squares. And we're going to talk about how Punnett squares let you see these different possibilities. Before we go on, this is the symbol for female. This is the symbol for male. When you're looking at genetic crosses, one of the things that you might see are these symbols to show you which organism is the male, which organism is the female. For now, that's not important, but we're going to see that you can, for the male, make one sperm, and if that sperm fertilizes an egg cell, it can contribute the allele that it carries. So one possible sperm that you could make from this precursor cell would have the dominant allele. That dominant allele would be given to any fertilized egg cell that sperm cell fertilizes. Or that precursor cell could make a gamete that has the recessive allele. If that happens, then the offspring, the fertilized egg cell, would have that recessive allele. Now let's look at egg possibilities. So one possible egg that could be made would be one carrying that dominant allele. Based on whether or not that egg is fertilized by another sperm, that determines what alleles will be given to the offspring. Same thing with the other possible egg we could make, which would have the recessive allele. And then we can see what would happen if that recessive allele was in the egg that was fertilized. So another thing I want to point out, This is showing you the probabilities of having offspring of a certain genotype. What this means is that you have a 1 in 4 chance of having homozygous dominant. This you have a 1 in 4 chance of having this combination where it's homozygous recessive. You have a 1 in 4 chance of getting the dominant allele from the father and the recessive allele from the mother, and a 1 in 4 chance of getting the dominant allele from the mother and the recessive allele from the father. Notice in both of my heterozygotes, no matter what the source was, I always write the dominant first. So now we're going to start looking at some of the Mendelian genetics. So the first sets of experiments that Gregor Mendel did were monohybrid crosses. He looked at one trait at a time and he looked at inheritance patterns. So the first trait that he looked at, one of the kind of more famous ones, is round versus wrinkled. And what he ended up finding through his crosses is that if he took a purebred wrinkled seed plant, and a purebred round pea plant and bred them with each other, all of the hybrids in the F1 generation were round. Because, as we now know, all of them were heterozygous. Then, if he took two of the hybrids and he mated them with each other, or he self-pollinated a plant, that produced a very different F2 generation. That recessive trait that seemed to disappear in the F1 generation is back now. And out of all of the offspring that were formed from these unions, and he collected a lot of data. He would have like 1,000 plants that he would look at the results from. He would find that there was a 3-to-1 ratio of round to wrinkled. So for every 3 round pea plants that he found, he would find 1 wrinkled. That doesn't mean that every single time this cross happened, he would produce 3 round plants and 1 wrinkled plant. No, it just means that the probabilities worked out to 3 out of 4 times it was a round plant that was produced, 1 out of 4 times it was a wrinkled plant that was produced. So now we're going to look at this in terms of probabilities because later on math is going to get helpful for us. So let's erase the genotypes and let's now look at the probabilities. So you have a 1/2 chance of any given allele being given to the gamete. What that means is that you have a 1/2 chance or 1 in 2 chance of getting a particular allele from the green parent, the male, and you have a 1 in 2 chance of getting a particular allele from the female or purple parent. And that is found everywhere. Now, if you want to know the probability of both of those events happening, getting a specific allele from dad and getting a specific allele from mom, you need to multiply the two probabilities times each other. So that's 1/2 times 1/2, which means each of those probabilities has a 1/4 chance. There's a 1 in 4 chance of getting a particular allele from dad and a particular allele from mom. So you multiply probabilities to find that out. Now, if we're looking at the probability of being heterozygous, we have 2 possible outcomes that can make that. So what we do to those probabilities is we don't multiply them, we add them together. So the probability of either getting this combo to be heterozygous or the probability of getting this combo to be heterozygous is equal to the individual probabilities added together. So 1/4 plus 1/4, which equals 2/4, which equals 1/2. So that's why when you're doing these Punnett squares, you have a 1 in 4 chance of having a homozygous dominant. You have a 1 in 4 chance of having a homozygous recessive, but you have a 1 in 2 chance of being heterozygous. And that is all expressed over here The P represents probability, and the little subscript of either the homozygous dominant, homozygous recessive, or heterozygous is just showing you which probability we're looking at. And this is going to become important later because we're going to talk more about probabilities. So if you have an unknown organism and you're trying to figure out what its genotype is, what you can do is you can do a test cross. Now, obviously, this doesn't work with humans because you can't breed humans to see what genes, what particular genes a person has. Don't try it, you will probably get punched. But with plants or with animals, you can do this. So we have a flower of unknown genotype. It's got the dominant trait, which is purple in this case, but we don't know if it's homozygous dominant or if it's heterozygous. What you can do is you can cross it with a homozygous recessive individual. If all of the offspring have the dominant trait, then it's probable that both of the alleles that that unknown parent had were dominant. If half of the offspring have the recessive trait and half of the offspring have the dominant trait, then you know that one of the traits, one of the alleles carried by that unknown parent had to be recessive. Because the only way to see the recessive phenotype is to have two recessive alleles. And of course, it being AP, we got to complicate things. But luckily, you should have seen these your freshman year as well. Dihybrid crosses look at the results of crosses between individuals who differ in two traits. We're looking at two traits at a time here. So notice we've got round and wrinkled still, but we're adding another variable. We're adding color. So here we see that the dominant color is yellow and the recessive color is green. And still, the dominant trait is round and the recessive trait is wrinkled. So there are 4 different combinations you can have. You can have trait 1 be dominant, trait 2 be dominant. You could have both traits be recessive. And then you could have trait 1 be dominant and trait 2 be recessive. And you could have trait 1 be recessive and trait 2 be dominant. Those are your only possible phenotype combinations. Now, there are a lot more genotype combinations, but I'm going to show you 3 different methods for doing this. Method 1 is to do a giant Punnett square like this, and this gets more complicated because you've got to figure out the possible gametes you can make. So for example, this individual right here can either give a dominant G or a recessive G. But paired with that dominant G could be either a dominant W or a recessive W. Paired with the recessive G could be either a dominant W or a recessive W. And so you figure out which gametes can possibly be made, and then you've got to fill them in this Punnett square. So for example, everything in this first column right here has to have at least one dominant color allele, one yellow allele. Why? Because it's getting it from this gamete. And each one in this row is also going to have at least one dominant W allele, which it had to get from this parent, this gamete. This is complicated. This can be very frustrating. So it's not going to be the method that I recommend. However, it's good for counting out phenotypic ratios. Typically, if you have an F1 generation that is hybrids and you cross them, your F2 generation is going to have a ratio of 9 to 3 to 3 to 1. 9 that are all dominant phenotype, 1 that's all recessive phenotype. You'll have 3 that are dominant for one trait and recessive for the other, and another 3 that are recessive for the first trait and dominant for the other. Now, you've got another method you can do. You can do double Punnett squares. So let's say I have 2 individuals who are both hybrids. So they're heterozygous for, let's erase that. They are heterozygous for the first trait, heterozygous for the second trait, and we're crossing them with another double heterozygote. So a double hybrid, basically. Let's say we want to figure out what portion of these will be, let's say, what portion of their offspring are likely to be heterozygous for the first trait and homozygous recessive for the second trait. Well, what we're looking for is the probability that you'll be heterozygous for the first trait, and that's 2 out of 4, or 1/2. 1/2 of the offspring will have that heterozygous genotype. And then we want to see what's the probability that they'll get the recessive trait. Well, it's a 1 out of 4 probability. To find out the chances of both of these happening, we have to multiply them. So the probability ends up being 1 over 8. If you have trouble multiplying fractions, basically pretend like you have a fraction line extending across. You multiply across the top, multiply across the bottom. So that is a much faster way to do this. However, you can actually have a more complicated problem. Back when you had calculation problems on the AP test, I've seen a question kind of like this, where you had more than 2 traits and you were trying to figure out what proportion of the offspring or what probability of the offspring have a particular genotype. So for this, you can just do probabilities. So the probability of getting the recessive allele for that first trait is going to be half. Probability of getting the recessive allele for that second trait is going to be half. The probability of getting the recessive for that third trait is going to be half. And for that fourth trait is also going to be half. This is from the first parent, though. The probability for the second parent to give you that recessive trait is 1/2 for the A, 1/2 for the B, 1/2 for the C, and 1/2 for the D. This represents A, B, C, and D, recessive, 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 recessive. By the way, when you're listing out genotypes, if you're not calling them homozygous or heterozygous, please don't just say A, B, C, or D. You have to specify whether they're capital or lowercase. So I usually would say big A or little a. So you multiply all of these probabilities together, which is basically 1 over 2 to the 4th power, so 1 over 16 times 1 over 16. And when you multiply that, that's going to give you your probability of having all recessive for all of those offspring. And that is 1 over 256. So there's a very tiny chance of getting an offspring that's recessive for all 4 traits, but there is a chance. It's not impossible. So let's do one of our first of our 3 practice problems. This is going to be a dihybrid cross, but I like this problem because it's not just, figure out probabilities. So we have a man who has a dimple and brown eyes. His father had blue eyes and no dimples. Married a woman with a dimple and brown eyes, whose father had blue eyes and no dimples. What is the probability that their first child will be blue-eyed without a dimple? So we're assuming that the dimple is dominant and brown eyes are dominant. So first thing I'm going to do is I'm going to designate a letter for each of these traits. I'm going to say that dimples are going to be D. So we're saying the dimple is dominant, no dimple is recessive. And I'm going to do B for the eyes, and I'm going to say that brown eyes are dominant, so that's going to be the capital B trait. So the man has a dimple, so he has to have at least one dominant. We're not 100% sure what his second allele is. And he's got brown eyes, which means he has to have at least one dominant. We don't know what his second allele is. His baby mama has she has a dimple, so she has one dominant. Don't know what the second allele is. And she's got brown eyes, so she's got one dominant. Don't know what the second allele is. If you're like, oh, how do we figure out what the second allele is? Well, that's when you look at their dads. So both of them had dads with blue eyes. Blue eyes are recessive, and they had fathers with no dimples. So their fathers were homozygous recessive. Since their fathers had to contribute at least one of their alleles, they have to have a recessive allele for each trait. So now we need to figure out probabilities. What's the probability that their offspring is going to be a double recessive, double homozygous recessive? So what's the probability of them getting recessive alleles from dad? Well, it's going to be 1/2 times 1/2. What's the probability of them getting recessive alleles from mom? Well, it's going to be 1/2 times 1/2, which ends up becoming a 1/4 chance getting those combos from dad, 1/4 chance getting that combo from mom. When you multiply it out, it's a 1 in 16 chance. So that's the probability of them having a blue-eyed, dimpleless child. We cannot do test crosses for humans because I think that kind of genetic testing through breeding is probably against the law. And even if it's not against the law, it's definitely creepy. So what we often have to do is play guesswork or detective to figure out what genotype a human might have. We do this using pedigrees. So pedigrees will show the gender of a person. So usually if it's a square, it's going to be male. If it's a circle, it's going to be a female. If it is a filled in shape, it is going to be an affected individual. So they have whatever trait that you're mapping out. And if it's an empty or hollow shape, then it's going to be an unaffected person. They don't have the phenotype, but keep in mind, you can have what's called a carrier. And that's an individual who carries a recessive trait but does not express it or show it. So anybody who is heterozygous would be a carrier of a particular trait. So a couple of examples. This is a dominant pattern of inheritance. You'll notice that every single affected person has to have at least one affected parent. Dominant traits tend to appear in every generation when you have them. And also notice you have an equal chance of getting this, whether you're male or female. That's going to become important later. Now notice, when you have a recessive trait, the only individual who has this is this one young man. Now, what you have is they were able to kind of trace this back to a single individual where this trait originated. This is actually the pedigree for the last royal family of Russia. So that younger boy is, I think, like Prince Alexander. And this trait is hemophilia. So somewhere his great-grandmother ended up having a mutation for something that would cause hemophilia. She had 3 kids, and she passed on that recessive trait to 2 of her kids. They were both carriers. All right. Then they married unaffected individuals who were homozygous dominant, and it passed that trait on to their children. So here, 3 out of their 4 children had the trait, and 1 out of their 3 children had the trait. And this is why inbreeding is a bad thing, because those 2 cousins married, and one of their offspring got a recessive from each parent, which means they had this disease. In this case, that was hemophilia, and hemophilia can be life-threatening because with it, you get a cut, and you just keep bleeding. Your blood doesn't clot. All right, now we're going to look at some other patterns of inheritance. Incomplete dominance is when you don't have the dominant trait completely cover up the recessive trait. Instead, you'll have an intermediate phenotype for the heterozygotes. Flower color will often have something like this. So in that case, you have 3 different phenotypes. You have a dominant, a recessive, and a heterozygote phenotype. Codominance is where you have more than one dominant allele that will show up when it's present in a heterozygote. So the best example of this is the ABO blood group, though there are some other examples of this that we may encounter in test questions. And this is kind of exemplified by people who have type AB blood. What this basically means is this is a particular antigen or molecule on the surface of blood cells. If you have type A, you have a specific one. If you have type B, you have another one. If you have type O, you don't have any antigen. And if you have type AB, some of your blood cells will have the A antigen, some of your blood cells will have the B antigen. If you have type A blood, there are two different genotypes you can have to get that. Sorry for the zooming. You can either be homozygous dominant or you can be heterozygous with a recessive allele. If you have type B blood, you can either be homozygous dominant or you can be heterozygous with a recessive allele. You can also be heterozygous to have both dominant traits, in which case you would express both. They're co-dominant or dominant with each other, and you have another phenotype, which is a combination of the two. And then finally, you have your recessive phenotype, which is the homozygous recessive one. I tend to like to write this where instead of writing big I-O, I'll write a lowercase I, which helps me remember the recessive. There are also other places where I've seen instead of writing an I with a superscript, they'll just have A, B, and O. I don't like doing it like that just because you forget what's dominant and recessive, but you can do it whatever way works better for you. And let's see if I can get to the next slide. All right, so now we have another practice problem that we're going to do with blood type. So we're going to say that A and B are dominant over O, but A and B are co-dominant, and then we have some more explanation. But here is where the problem is. A man with type A blood marries a woman with type A blood. Their first child has blood type O. What are the genotypes of the father, the mother, and the baby? Well, the father has to at least have one A allele, as does the mother. And the child has type O blood, which means that they have to have two recessive alleles. Well, they had to get those recessive alleles from someone, so we can backtrack and say, okay, both of that child's parents were heterozygous. Fairly easy. There are, of course, more complicated ones that will show up on the AP test. By the way, what do you notice about this pedigree that's a little different from all the others? Which individuals have the trait, whatever this trait is? You'll notice it only affects males. There are some traits that are actually sex-linked. What that means is they're linked to gender. The reason for this is because in humans, and in a lot of organisms, you have different chromosomes that determine your gender. In humans, we have X and Y, although other organisms can have other chromosomes. And I mean, there's a lot of complicated stuff that we're not going to talk about here. So if you are a female, you have two X chromosomes. If you are a male, you have one X and one Y. Unfortunately, the Y chromosome is the smallest chromosome. It's missing a lot of genes that are found on the X chromosome. So because of that, males will sometimes have only one copy of a particular gene. It will be the copy that's on their X chromosome. They don't have a second copy on their Y chromosome. So they are more likely to display the recessive trait for something that's on the X chromosome. So a few of these that are more common in males than females because they're on the X chromosome are hemophilia, color blindness, and male pattern baldness. And there are traits that males can inherit that females don't because they're on the Y chromosome. But let's do a practice problem. So we have a cross between a white-eyed female fruit fly and a red-eyed male. We want to know what percentage of the female offspring will have white eyes, and we're told that white eyes are X-linked and recessive. Now, the way that we usually represent chromosomes when we're doing sex link, we have to indicate what's an X and what's a Y. So we're going to say that the female fruit fly is XX and the male fruit fly is XY. Okay? But we still have to indicate that allele, whether it's dominant or recessive. So the way we indicate that is the dominant trait of red eyes is indicated with that superscript of capital R. The recessive trait of white eyes is indicated with a lowercase r. And then your third possible chromosome is the Y chromosome. So this is our cross. We have a female who is white-eyed. She has to have both recessive copies. And the male is red-eyed. Well, he has to have at least one dominant. He's only got one X chromosome for it to go on, so that has to have the dominant trait. So, what percent of the female offspring will have white eyes? Well, we can do our trusty old Punnett square. And if we do that, we'll see that we have a bunch of red-eyed girls and a bunch of white-eyed boys. So these are the girls. They're all red-eyed. And these are the boys, and they're all white-eyed. So their chance of having a female with white eyes is 0%. And yes, you can have a 0% chance of having a certain offspring, depending on the genes. Interesting case about X chromosomes that I just like to point out is that actually in any female, one of your X chromosomes is usually condensed and inactivated. It's called a bar body. You can actually see it in some microscope images. So this is a nucleus. That's the bar body. And this process is very random. So you have a fertilized egg cell. In like half of the cells, you'll have a particular X chromosome inactivated. In the other half, you'll have the other X chromosome inactivated. And this is responsible for the trait of calico cats. They have patches of different colored fur. So what happened is that in this cat right here, I'm going to switch to the laser pointer. In this cat right here, you see some kind of orange fur, and then she also had patches of kind of black fur. So one of her X chromosomes had the gene for black fur color, and it was expressed in certain parts of her body, and another one of her X chromosomes had the gene for orange color, and it was expressed in other parts of her body. That is one of my former cats. She was a calico, and because of this, I'm going to say like 99% of calico cats are females. I have seen rumors of calico males, but I've, like I said, rumors. All right, so that is our long reintroduction to genetics for y'all. We'll do practice problems.